Good afternoon. We're starting our last session. Couple of minutes.
Var mı?
Our last speaker today is Professor Ogün Adebali from Sabancı University and also from our institute, from the Institute of TÜBİTAK Fundamental Sciences Research. It's going to do a speech on the title is Exploring Protein Variants and Associated Disorders through Phylogenetic Approaches, topic which is very close to what we will study in the past few days. Nereden bağlı?
Aşağıda şey kaçmış olabilir diye.
Okay, hello everyone. So I think it was a long day for you. So I hope this presentation will inspire you regarding how to analyze protein variants in context of phylogenetic approaches. So this talk is not going to be a regular training, but I hope you will get something to learn about with that. So it is going to be similar to a seminar, but if you have any questions, please let me know. I will give more insights regarding the methods and the approach that we use. So in my research group, we focus on 3 stages of mutagenesis: before mutagenesis, during mutagenesis, and after mutagenesis. So before mutagenesis, we are interested in DNA damage and repair. So we analyze genomic maps of DNA damage and repair. Perimutagenesis means that we try to keep track of mutation profiles, because if you analyze what kind of mutations occur in an organism, you can have an idea regarding the mutagens, basically. What are the mutagens? Some mutagens will have some footprints, and it would be possible to track them. And the post-mutagenesis, after mutation occurs, what are the effects of the certain mutations? So in that case, we depend on some algorithms. We develop some algorithms. So today I'm going to talk about post-mutagenesis, how to understand and interpret the effects of mutations. So we benefit from evolutionary data because to me, it's like the longest running experiment. So today what we see in the genomes and what we do not see in the genomes give important insights regarding the function of the protein. So what are the critical amino acids that are important for the function? And based on the evolutionary conservation, we can have an idea regarding their potential effects on disease. So in this talk, I'm going to talk about, by using phylogenetic information, can we predict the effects of protein variants? Can we infer co-evolving positions? And finally, can we understand the role of each amino acid on a protein? So let's start with the first one, predicting the effects of amino acid substitutions in disease context. So if you look at human genome, each human has approximately 25,000 variants that are in the genes, and 9,000 of them are missense mutations. Missense mutation means that they cause an amino acid substitutions in a protein. And 400 of them are rare mutations. It means that we have no idea regarding what they do. So are they pathogenic? Are they neutral? So we have no idea. There is no literature about that. And in the population, the frequency of those variants are very low that nobody has studied. If you look at the pool of all theoretical variants, which is 9 billion, you can think about 3 billion letters in the human genome and 3 letters that it can turn to, one position can turn into, 9 billion variants. And what we know about them is only 5,000 of them. We have some idea regarding what they do. And the rest is an ocean, basically. We have no idea. So what we can do, right? So we can go and do experiments, biochemical experiments, but it would be infeasible to do that. Or you can go and look at the literature, only 500,000 of them have some value. But regarding the rest of them, we depend on computational tools. Imagine that we have a patient that this child has a mutation, aspartate to tyrosine mutation. So you are interested in whether this substitution is a pathogenic or neutral. So what we can do is we can benefit from multiple sequence alignments. So we can have a multiple sequence alignment. We can get the homologous sequences from all the primates, mammals, vertebrates, invertebrates, sometimes plants, and even bacteria. We can look at the conservation. And if it is fully conserved, then it means that it has a critical function, then this mutation would be pathogenic, right? So I will give you some scenario here. So in this case, the first scenario, if you look at the human, it is aspartate. Let's say chimpanzees are aspartate, birds, fish, insects, they're all aspartate. There's full 100% conservation. In that case, it means that this aspartate has a critical role. Then when you have aspartate, the tyrosine substitution, it is probably causing something bad, and you can categorize it as a pathogenic mutation. Let's look at the second example. In this case, you have one mutation in one of the birds, let's say. This is still one single organism that is not conclusive to say that it's neutral, because this substitution could have occurred with another compensating substitution in that organism, right? So by just looking at single tyrosine, it's not possible to say that it's neutral. So you need extra evidence for it. Compared to the second scenario, if you look at the third scenario, if this aspartate substitution was in a closer species, it is still one species. It is still not conclusive. But relative to the second scenario, I think we should give more weight to the neutrality score in the third scenario, because it is a closer species, so that the neutrality score that we can interpret should be higher, right? So therefore, the species is important, and the closeness to the query species, the human in this case, should be considered. If you look at the fourth example, we have two different tyrosines. Relative to the first and second scenario, we have two independent events. Then we can say that, okay, we should give more neutrality score to this tyrosine. If you look at the fifth scenario, we have 8 of them. And when you have 8 of them, you can say that, okay, throughout millions of years of evolution, 8 times you see tyrosine, then you conclude that probably it's a neutral substitution. D2Y substitution should be neutral. However, if those 8 substitutions were dependent, just like here, if 8 substitutions occurred in the common ancestor of a certain clade, in this case, this clade, it happened here, obviously. Remember about the compensating mutation. So when this substitution occurred, a compensating substitution could have also occurred in the common ancestor of this clade. So to me, rather than the number of tyrosines in the multiple sequence alignments, number of this substitution, number of events are important. So multiple sequence alignments, if you only use multiple sequence alignments, this evolutionary relationship of those tyrosine substitutions are invisible to us. But if we use phylogenetic information, if we align the multiple sequence alignment to them, then we can pinpoint where this substitution occurs. And rather than counting the number of tyrosines, we should count the number of the events, how many times this substitution occurs. So this is the problem of multiple sequence alignment-based approaches. So therefore, I think we should use phylogenetic tree as a piece of information in that case. So we have developed an algorithm called PHECT. PHECT stands for Phylogeny-Aware Computing of Tolerance. I will give you a toy example here. So imagine that this is our phylogenetic tree, and our human sequence is here. So what we know is today, the probability of these are 1, because these are what we observe today, right? So we have T's here, we have I's, we have also T here. What we do not know is the common ancestral states. So we do not know what this common ancestor was, but we can infer in a probabilistic manner. So we can say that, okay, probably this was 1, the probability of T was 1, but when we come here, the probability of T is not 1 anymore because of the isoleucines here. So 0.86 here, 0.11 here, right? So we can assign probabilities to each node for each of the amino acids that we can have, which is 20. So after doing that, we are interested in counting how many times this substitution occurs, right? But since we cannot be certain about the states at the nodes, we have probabilities. What we do is the following. We start from query species. We go to the upper node first, and we note down the probability changes. Here, there is no probability change. It was 1 here, and again, T is 1. And here, there's no probability change. If you look at dashed lines mean that there's no probability change. But when we come here, the probability of isoleucine increases from 0 to 0.11. And if you go more, isoleucine probability here goes from 0.11 to 0.96, so it increases by 0.85. So in each branch traversing, we note down the positive probability difference, and then we prepare a table, right? And after that, we also take the weight of the distance, the weight of the node. For example, the change here will have more effect, more weight to the neutrality score than the change here. So there is a function that we apply, which is inversely correlated with the distance. If the change is in a closer branch, we give more weight to that. If it is in a far branch, we give less weight to that. And after that, we have a tolerance score. So if you look at the performance of PHECT and compare it with the other tools, other statistical tools mostly, the red line, this is the AUC curve. I think you have went through the deep learning class, so machine learning class. Probably you are familiar with that. So if the area under the curve is large, it means better performance. So if you compare this one with the most famous tools like SIFT and Polyfan, so it is performing much better. And if you compare it with the others, and Eve is very popular and recent, so the negative value means that PHECT is doing better than the others. So it worked fine. So the reason why it worked fine is the following. I will give 2 examples. One example is, for this substitution, arginine to histidine substitution, the true label is neutral. But SIFT thinks that it is pathogenic, and PHECT thinks that it is neutral. Why is that? If you look at here, the only histidines are in the lemur and green anomaly. So the rest is arginine. So SIFT only looks at the multiple sequence alignment, and since it sees only 2 histidines, it considers that it's minority. Therefore, it categorizes histidine as pathogenic because it's only 2. But what PHECT does is the following. First, it considers this as 2 independent events, and it considers this as an important indicator for neutrality. And second, in lemur, histidine substitution occurs. And if you know what lemur is, it's a primate. Therefore, it is very close to human. Considering these 2 factors, PHECT thinks that it is neutral and the true label is neutral. The next one is about a pathogenic mutation. So here we have a pathogenic mutation I to L, and SIFT and polyphen thinks that it is neutral. And if you look at multiple sequence alignments, isoleucine is the brownish one, the blue one is leucine. There are many, many leucines in the multiple sequence alignment, and SIFT and polyphen use multiple sequence alignments. Therefore, they think that this should be neutral. However, if you look at this clade, they are all isoleucines. Therefore, since they are far away, the distance is large. Therefore, FACT reduces their weight, and the number of times you see a change to leucine is very low. Therefore, FACT thinks that it should be pathogenic. So, number of leucines in the multiple sequence alignments, although it dominates, it doesn't matter. What matters is the number of times that substitution is seen. So there are some advantages of FACT. One advantage is it is species independent. So it is not optimized for humans. So you can start traveling the tree from another species like mouse, and you will get a neutrality score for mouse, which is a quite important benefit. The other one is interpretable single score. So this is not a black box. We know exactly why FACT gives a neutrality score that it considers. So it is not a machine learning, therefore it's fully interpretable. So it is open for development, high accuracy and precision, and it also offers some promising machine learning features. So what do I mean by that? I will come to that, but this has been published and it made to the cover. So this scientist represents the algorithm, and this path represents the phylogenetic tree. Basically, algorithm travels on this path and notes down all the substitution. So this is the representation of the algorithm. So then we said that, okay, so we have a nice working statistical tool, but can we use its output as an input for machine learning model to have a better performance? And we took the output of FACT and used them as an input for a machine learning model. But not only that, but also other features coming from multiple sequence alignment, phylogenetic tree, ancestral sequence reconstruction, and amino acid properties. So basically, the idea is following. The machine learning model that we apply is XGBoost, and it is a version of a decision tree. So here is a representation of a decision tree, and this tree is fed by a phylogenetic tree. So the roots are basically phylogenetic tree. The output is a decision tree. So first we compared FACT with its foundations to show the effect of all of the features. So I will not go into detail that much, but what is important here is we have also provided the tree boost, MSA boost, and FACT. But what I would like to pinpoint here, FACT boost is much better than FACT, especially for hard cases, which is TS4 and TS5. TS means test set. Test set 4 and test set 5, the difference is huge. So there is definitely some improvement with the machine learning. And we compared ourselves with other tools. So we performed better almost in every case, but I would like to pinpoint again TS4 and TS5, which are the hard cases. You see a huge improvement in the performance because, especially in the hard cases, the importance of phylogenetic tree basically rises. So what are important features in this machine learning model? So the first and very important feature is the FACT output, which is not surprising. But what is also important is if you look at the next four, so the first 5 features are all tree-related features. So therefore, rather than multiple sequence alignment features, which are also important, they are contributing, but first 5 features are coming from tree features, so which are sort of an indicator of the importance of phylogenetic trees. I'll show you the performance with 2 examples again. So here the true label, in this case E to K substitution, is neutral, and FACT correctly predicted it as neutral, and all of the important tools say that they are pathogenic. The reason is, again, the following. There are 2 independent events here. One of them is very close to human, therefore FACT thinks that it is neutral. The opposite example is about this case. So FACT thinks that it is pathogenic, and indeed it is pathogenic, So if you look at other tools, they say that they are neutral. The reason why fake boost thinks that it is pathogenic is the following. See the conservation of this clade. They are all almost identical to each other. There's a single line, right? So there's no diversity in this clade. So what matters is whether you see any leucine here, which is the alternating amino acid in this clade. And if you do not see that, then probably you will categorize it as a pathogenic, and fake boost exactly does that. And while we were doing that, Google DeepMind company, so they actually had a breakthrough with AlphaFold. You remember that, right? So when we were preparing the manuscript, they came up with a tool to predict the effects of missense mutations. And then we compared ourselves with alpha missense. And again, look at the comparison of fake boost with alpha missense. So there is better performance for fake boost, although it is. But if you look at the hard cases, the improvement is 20%. So which is quite satisfactory in our opinion. So we have a tool that performs better than DeepMind's prediction tool. We also compared ourselves with other tools with another data, which is coming from deep mutational scanning. In this experiment, what they do is they mutate each and every position of a protein with all of the potential 19 substitutions, and they measure the protein activity. So in this case, we are not measuring the pathogenicity and neutrality, but we are measuring the effect of mutation on the activity of the protein. And in that case, we look at the correlation. And in that correlation, we are performing similar to alpha missense and CPT1, and we are better than EU and Bravo. This also in molecular biology and evolution. But I always talked about our successes, but where we fail is also important, and it can teach us about the data. So in this case, you are seeing two different ways of submitting a ClinVar pathogenicity. So clinicians, what they do is they submit a pathogenic variant to a database when they are sure that the variant is pathogenic, right? But there are two ways of doing that. One is by following the guidelines. The other one is not following the guidelines, right? So normally this is the distribution of the pathogenic variants. So this amount of pathogenic variants were submitted through guidelines, and these were submitted without guidelines, right? But if you look at where we fail, which is around 460 or 70, so the number of guideline submitted pathogenic variants is much lower than the background. So it suggests something. What it means is maybe the data set that we use is not providing the gold standard. Maybe the labels are also not exactly 100% correct. So this data show that we should be careful about the gold standard data, which is not necessarily true all the time. The other case where we fail, and this is the most important slide in my opinion, and most exciting one, I'll tell you why. So this is glycine to tryptophan substitution. So if you look at the number of tryptophans, so there are 3 of them, and since there are 3 of them, fake boost thinks that, and 2 of them are very close to human, and fake boost thinks that it is a neutral substitution. And it is a pathogenic substitution, and it is in the clinical reports, it is reported as pathogenic. How it can be, right? When I look at that, and when I see 3 independent tryptophans, I was sure that it must be a neutral one. However, it is not. Why is that? If you look at another position on the same protein, wherever you have tryptophan, in that species, you have a change in another position. In this case, it is alanine. In this case, it is also alanine. In this case, it is threonine. So basically, it means that tryptophan is only allowed when there is a substitution in another position. So this suggests coevolution, right? When you look at the protein structure, they are close to each other. So somehow this tryptophan is compensated by another mutation in the neighboring position. So therefore, since we evaluate the positions independently, maybe it is not the right way. Maybe we should be evaluating the positions together, right? So this is something that we should take into account. But this brings me to the second part of my talk, which is about coevolution. Somehow it's not reflected on the Zoom yet, but I'm not sure, maybe. Okay, here we go. So we have to find the coevolving positions, but it also suffers from the same problem of the multiple sequence alignment. So we don't know, if we do not have any information regarding the phylogenetic tree, we don't know where these substitutions occurred. So here I'm giving you two positions in a multiple sequence alignment. So these two positions can have two different evolutionary scenarios. One of them is on the left, the other one is on the right. On the left one, maybe these substitutions occurred in the common ancestor of that. It means that this occurred only one time, A and D substitutions. But the right scenario might also be possible. Maybe these substitutions occurred independently. In this case, which one should have higher coevolutionary score is the question. And it is simple, of course, it should be the one on the right, because 3 times you see the co-evolving site, right? But if you do not have any phylogenetic information, you cannot infer it. Therefore, we need to take that into account. So to give you a better example, I'm still waiting for slide to change. Oh, internet connection is not stable. Let's see. Okay. So if you look at this multiple sequence alignment and two positions, we are interested in the number of times these two positions have parallel substitutions. And you see there's 1, 2, and 3. So this suggests a high coevolutionary signal here, right? So we want to find that. So what we did is the following. So remember FACT. So FACT can categorize the tolerable and intolerable substitutions. And if you do that, I think we should be, rather than using 20 amino acids, we should be using only the tolerable and intolerable ones to reveal the coevolution signal. So what we do is, in this case, imagine that T is tolerable and R's are intolerable. What we do is we assign 2 letters for the tolerables, 1 letter for tolerable and another letter for the intolerable one. And we do the same for other positions. Then we have a clean coevolution signal. And phase gets multiple sequence alignment, phylogenetic tree, and ancestral reconstruction as inputs. And then it gives coevolution scores for all position pairs. Not only tolerable, intolerable categorization, but also gaps in the multiple sequence alignment are important. And then we simply we calculate the probability changes just like in FACT. We travel through the tree, we note down the probability changes, and we have a table. After we have a table between 2 positions here, we look at the correlation. So if they are highly correlated, it means that 2 positions change at the same time throughout the evolution, right? So what is the correlation metric? It is concordance correlation coefficient rather than Pearson correlation. So if you look at concordance correlation coefficient, it gives more score to the correlation trend that is close to the x equal to y line. For example, Pearson and correlation coefficient concordance correlation coefficient CCC is 1 here, but here Pearson correlation is 1, but CCC is 0.82 in this case. Although they are perfectly correlated, it is a bit far away from the x equal to y line. So we use CCC and it worked out nicely. But how do we test our data? The first solution was create a database from experimental sets, but it didn't work out for us because in experiments, you're only measuring certain activities. So it doesn't necessarily reflect the evolutionary signal. But what we can do is we can benefit from protein structures. We can assume that if two amino acids are close to each other, they are more likely to co-evolve. Although it's not necessarily showing coevolution, but they are more likely to evolve. So basically the idea is following. We should be able to give more coevolution score for the close ones. In this case, they are shown in purple. And for the far ones, we should give them less score. So the idea is to be able to separate between these two distributions of the close ones and the far ones. And it worked out. So FACE is performing better than other coevolution tools like DCA, Gauss DCA, and so on. So AUC is quite nicely around 0.9. So to give you an example, so in this case, there's no, although this is fully conserved, other coevolution tools give high score to them, but FACE gives -1, which shows no coevolution. Basically, there's no change here. Therefore, FACE gives them negative value. There's no coevolution because although they are fully conserved, they do not change at all. And if you look at this example, it is quite striking. If you look at these multiple sequence alignments, they are very messy, and I don't see any parallel changes here, right? But if you clean it up by assigning two letters for tolerable and intolerable, you see here they are nicely co-evolving, right? So therefore, we give high score to that. Since the other tools do not consider about the tolerable, intolerable discrimination, therefore FACE is performing better for this case. So now can we use these two things together, which is an unpublished data I'm going to show you. So first, we need to eliminate the coevolution contribution to the neutrality score, just like in the example of the tryptophan, right? So first, we thought that maybe we should be finding all co-evolving positions in a protein. And then we can check the change status on each branch for co-evolving positions, and then we can adjust the weight of them. I will give you an example. Imagine that we are interested in the neutrality score for position 1, and position 2 and position 3 are co-evolving with position 1. And in this branch, this reflects the change, total change, and this is the total change, and there is no change in branch 2. This much changes in branch 3 and branch 4. If you look at branch 4, for example, position 1 and position 2, they did change together. So it is a coevolutionary signal, right? What we can do is we can reduce the effect of branch 4's contribution to the neutrality score because it is a coevolutionary branch. So we reduced it to 1 because there is a perfect match. And in this case, this neutrality score should be contributed by this branch fully. So we adjust the weight. We didn't touch the weight, so it is 1. But here, since they are very close to each other, 0.9 versus 1, so we reduce it to 0.1. So therefore, we reduce the effects or contribution of some branches, which we see coevolutionary signal. I will give you an example of why this is important. If you look at this pathogenic variant, and it is reported to be pathogenic, and FACE thinks that it is neutral. But if you include FACE into account, neutrality score drops 0.03, which is a pathogenic score. Why? If you look at the 7th position, which is the main position we are interested in, and you see many times the alternating amino acid in green, so these dashed lines show the green amino acid, which is the A. So naturally, FACE thinks that it's neutral. But whenever you have an A here in the co-evolving position, you have another amino acid. Therefore, when you reduce the effect of branches or nodes of those changes which are providing coevolutionary signal, then your score becomes 0.03. Then you can categorize it as pathogenic, as it should be. If you look at this example, it is also similar. Normally, it is pathogenic. PEG thinks that it's neutral. But the reason why it thinks that it's neutral is because of some changes here. But whenever there's a change here, there's a compensating change. Whenever there's a change here, there's a compensating change, and same here. So when you reduce the neutrality contribution of these branches, your score becomes 0. Therefore, you can truly categorize it as pathogenic. And in the final part, I'm going to talk about how to understand the functional roles of amino acids. So gene duplication is a method in the nature that we have seen many times for innovating a new function. Imagine that this was an ancestral gene and it is duplicated. Now we have 2 copies. And each of these diamonds, let's say that they reflect a subfunction. So what can happen is the following. One copy can become a pseudogene. It means that it is non-functionalization. It can get deleted or it can accumulate many mutations, and it loses the gene status. The other possibility is subfunctionalization. Some of the subfunctions are retained in one copy. Some of the subfunctions are retained in the other copy. Now they are not equivalent to each other, right? And sometimes we have neo-functionalization, meaning that some function is changed in one of the copies. Then they are not the same protein anymore. They are not the same gene. So therefore, this is one way to innovate a new function. And orthology definition is not sufficient. I will give you one example. Orthology means that the homologous genes shared homologous genes due to speciation is called orthologs, right? So if you look at this human version and this chimpanzee version, they are related to a speciation, therefore they are orthologs. But this one and the other chimpanzee one also, they are orthologs because of speciation. But if you look at this one, there's a huge divergence of this purple protein. Therefore, what I would do to consider functionally equivalent orthologs, I will take this one and make it equivalent to the human version rather than this one. So therefore, this is important to identify which of the paralogs, which are here, these are the paralogs, are the true ortholog of the human. And we have a pipeline, which I'm not going into detail. And after this pipeline, we can put the functionally equivalent orthologs in the same bag. Therefore, we can do sensitive analysis on them. And we have used G-protein coupled receptors. They are quite important with respect to being drug targets, because 1/3 of the FDA-approved drugs target for these GPCRs because they are at the cell surface. They are important in signaling. They recognize many different ligands. They initiate signal transduction pathways. But their evolution is very, very complex. The reason is the following. So you have one receptor, you copy and paste that receptor, tweak it a little bit, have some mutations, a bit of mutations, then it recognizes a new ligand, then it's a new receptor, right? So copy, paste, and change it a bit, and then it's a new function. And evolution used that method many times. Therefore, the evolution is a mess, and it is very, very complicated. But it's a good family for us to study because we like challenge and we like performing evolutionary studies. So the idea is following. What makes receptor A is the question, and what makes receptor B? So we take all the true orthologs of receptor A, all the true orthologs of receptor B, and we look at the conserved ones. Some conserved positions are shared between receptor A and receptor B. We call them consensus. So they are important in folding and main mechanism of GPCRs and so on. But there are some specifically conserved residues which make receptor A. They are shown in red here. They are conserved in receptor A, but they are not conserved in receptor B, or they are conserved in receptor A, but they are conserved to something else in receptor B. And same goes for the B-specific ones. So after we identify that, we realize that they are mostly in the ligand binding sites because they generally sense different ligands. Sometimes they are in the dimerization sites, and sometimes they are in the interface of the G protein coupling, which are important for starting signal transduction pathways because they are coupled to different G proteins, if they are. Right? Again, we have a methodology here. I would like to show you the last part, which is quite critical. I think I have a better version of that. So imagine that this is class-wide conservation. So the y-axis is showing, for example, receptor A conservation. And the x-axis is class-wide entropy. It means that the conservation should be high for these selected ones, but the conserved amino acid should be different across subfamilies. What I mean is, if the conservation is high for A and B, and if it is the same amino acid, they are involved in core functions, which is the yellow ones. But if they are conserved to different amino acids, they are important for receptor A and receptor B, but they are conserved to a different amino acid. Therefore, they are important in selective functions, right? So this is what makes receptor A, and receptor B, right? When we did that, we have looked at olfactory receptors. And then we have realized that their selectively conserved ones are located here. And we directly said that it must be the ligand binding pocket. And when we did that analysis, there was no structure of olfactory receptor. We got excited. We said that I picked up a phone and called my colleague. I said that we must have a crystal structure of that because we have a good hypothesis it must be a ligand binding pocket. He got very excited. We were looking for some vectors to order and so on. 2 days later, the first structure of olfactory receptor was published in Science. So it was a bit disappointing, but it was also satisfactory because the exact ligand binding site was here, exactly where we predicted. So without having any experimental data, with computational analysis, you can have a really accurate insights regarding biology. This similar story, this time it was not disappointing. A colleague asked us whether we can have some insights regarding some frizzle receptors. We have predicted cholesterol binding sites, and we shared the data with them. It was through Zoom because they were in Sweden. And they got shocked because exactly the cholesterol binding that they observed was the place that we predicted as cholesterol binding. So then we published this together. So two data had an agreement with each other, then it was a stronger message. And then I'm not going to detail, but this is also a study where we figured out the G protein coupling selectivity, and we identified that selectivity is not only determined by the interface, but also some networks in the core and in the upper side of the receptor, which was an important study to understand the GPCR activation. And the final story is about calcium sensing receptors. So we studied calcium sensing receptors in class C GPCRs. So look at here, calcium sensing receptors are here. They are highly conserved. But if you look at the neighboring plate, they are calcium sensing receptor-like proteins. But if you go to a database like UniProt or NCBI, you will see some of them annotated as calcium sensing receptor. However, they have nothing to do with calcium. And you can obviously understand by the divergence of that clade. They are involved in other functions. So therefore, it is very important to identify, okay, what is sensing calcium? It is basically this clade, but definitely not clade. Not this diverse clade. So we can now represent those subfamilies by applying profile HMS in the Markov models. And the Markov models are important in representing protein families. Basically, for example, you have this logo coming from multiple sequence alignment. There are some conserved amino acids. And what we can do is we can specifically build a profile HMM for this target by increasing the specific ones, the increasing the weights of specific ones and decreasing the weight of consensus ones. And you see we have a model that is specific to the clade of interest. So the idea is, rather than building phylogenetically each time, maybe we can have a profile HMM to scan for what is calcium sensing receptor, for example. This was the idea. So after we have this one, it worked perfectly. So our specific, subfamily specific HMM hit all of these target sequences, but it did not hit the neighbor sequence. So it was a perfect model for our subfamilies. And if you look at physiological evolution in calcium binding sites, so since in the experimental stage they overdose with calcium, the calcium levels are highly concentrated, we have 4 different calciums here. But which one is real calcium that is important to start signaling is unknown. So what we can do is we can have a specificity algorithm we can implement it, which I'm not going to into detail. We realize that the specific amino acids are enriched at around one of the calciums. And we think that this is the calcium that is required to trigger the signaling mechanism. So with experiments, you cannot figure out that. So you have to do some computational analysis. So calcium sensing receptors are extremely conserved. So this is the conservation branch lines, and these are the branch lines of other membrane clades. And a bit of biology. When calcium increases, active CASR inhibits this pathway, and when it inhibits this pathway, the calcium levels in blood is reduced. When there is low levels of calcium, inactive CASR activates this pathway and it increases the levels of calcium. But the problem is there are some loss of function and gain of function mutations in that protein. So what happens is, regardless of the presence of calcium, if you have gain of function mutation here, you have always active CASR. It means that you have reduced levels of calcium in blood, and it's called hypocalcemia. And if you have loss of function mutations, the CASR is always inactive, so this pathway is always active, and you have an excessive amount of calcium in blood, then it causes hypercalcemia. Now, this case is different. So, 2 opposite consequences of different types of mutations. So if you look at evolution and conservation, they are both going to be very well conserved, right? So because in either case, they cause something bad. Therefore, evolution and conservation is not sufficient to discriminate between loss of function and gain of function. What we can do is something else. Again, we benefit from evolutionary data, but this time we give the model not only calcium sensing receptor evolution, but also the evolutionary trends of other neighboring clades. So then we have a classifier, and we predict loss of function and gain of function mutations. So we have an AUC levels of 0.8, AUPR levels of 0.9, so it performs really well. But how is it possible? And if you look at the most predictive feature, it is the conservation score of one of the taste receptors. And why is that, right? So I will tell you why. In regular proteins, gain of function mutations have no effect in general, because a bit of overactivation for protein doesn't cause very bad consequences, I would say. So for CASR, it is not the case. So a bit of activation is causing something very bad. Therefore, the ones that are in taste receptors that are causing a bit of activation are seen in taste receptor. But the ones that cause loss of function in taste receptor that would also cause loss of function in calcium receptor, they are shared between calcium sensing receptor and taste receptor. This, I think, makes a difference. So we can learn from the neighboring clades to see what is allowed and what is not, and we can transfer that information to calcium sensing receptor to be able to discriminate between gain of function mutation and loss of function mutations. So which is a quite novel approach, in my opinion, and this is also published. And then, if you have no idea regarding a mutation causing loss of function or gain of function or neutral, then at least you have a prediction method to have a bit of insight, and this is the map of gain, loss, and neutral predictions. So my conclusion is this one. So when you use phylogenetic trees properly, it gives a lot of insights regarding to biology. And these are the scientists and scientist candidates that we have in our group, and they contributed to the studies that I mentioned. And these are the acknowledgements. If you have any questions, I will take them. Yes, please.
Ayıp Karaman from Acıbadem University. Thank you for this inspiring work. I wonder, by using fact and dates, can you also predict the severity of pathogenicity, or you just say it's pathogenic or not? Because from Hungary to Church, we can say that it's highly severe, highly pathogenic.
Right.
But from polar to polar, I believe that I don't know physics very much, but it is changing. You know, in harmonious change, even if it is from polar to polar, it might be severe in some cases. I don't know. I don't have the information. It's not my main thing, but I'm always curious about hearing this kind of.
Yeah.
Thank you for work.
Thank you.
Inspiring talk.
Thank you very much. So it's a great question. So the idea is the following. For fact, yes, because the neutrality score correlates with the number of changes that you see, right? So definitely the higher score means higher neutrality and lower score means higher pathogenicity. For fact, it is the case. And in the multiple sequence alignments, you see that if a polar change is allowed, you would also see that in the evolution, right? So therefore, you will have some severeness effect, definitely for fact. But for fact boost, I'm afraid it is not the case because it's a classifier. You basically try to classify as pathogenic and neutral. So the score doesn't necessarily correlate with the severity, but with fact, which I like better because I know exactly what it is doing, so it correlates, yes. And you remember the DMS data, deep mutational scanning, we look at the correlation there. So we see a correlation of the severity in that case. So it also shows that, yes, we can do that. Yeah. Please.
It was a very strong presentation. I have two questions. The first question is totally related to the types question. You say you predict the tryptophan change as neutral,
right.
It's actually pathogenic. But we know from nature that the change of any amino acid to tryptophan is partly penalized, even Blossom score table, because we have some changes, amino acids from any kind to the tryptophan, and it is pathogenic in many diseases, especially
they're often penalized.
Yes, in neurological diseases, in ion channels. So maybe you are predicting false in the case of tryptophan, your tool. Maybe actually it's pathogenic, but your tool is predicting it's false, and maybe it's not related to co-evolving. Maybe just a single change is the result of the pathogenicity. That's my first question. Can you just?
So I couldn't follow. So tryptophan substitutions are more. They are often pathogenic. And
yes.
In evolution, we see them 3 times in that case, right? So the question is, why do we see them in evolution 3 times? So although there's a nice conservation of all others, but 3 distant cases, we see tryptophan. The question is why, and we could explain that with the compensating mutation in nearby, right? So I couldn't follow the question.
Maybe just change of one amino acid, any kind to tryptophan is the result of the pathogenicity, but not the other amino acids nearby.
Then I would ask this question: why it is not pathogenic in the species that we observe that change, right? There must be a reason for that. Yes.
That's what I'm asking for. And the second question, you showed some prediction results with comparison with DeepMind.
Right.
Just I'm curious, is it statistically significant? Did you try any bootstrapping approach to find any significance?
Yes.
Because just a single run, maybe you have a higher score, but
right.
It is statistically significant.
Yes, we have tried exactly bootstrapping, and we have we have a pool, and from that pool we select subsamples, and then yes, it was significantly different. Significant, statistically different. Yes. Are there any other questions? Yes.
Yes, so you change the fertilizer three times. And like similar example, they change like lines into a square like in one, they change this single iron on the double of its constant. Iron to so it's question.
A good question. Yes, they are same, and you can think on how they can be same because they have different amino acid properties. One is closer with respect to physical chemical properties, the other one is not. So this is our approach. If they have similar tolerance throughout evolution, then it is expected to have 3 times no matter what. So if it is something severe, we would expect them to see rarer than the other one, right? So what we think is amino acid properties are important, but they are already reflected in evolutionary data. This is our approach. So amino acid properties are already in the data itself, so we don't have to take care of them independently. So that's why I said that evolution is the longest running experiment. Shall we check any questions? No, I think there are no questions on chat. Any other questions? Okay. So I wish you pleasant training sessions tomorrow and the other days. So I hope to see you again. Thanks.